This lesson is going to teach you how cells build proteins. So proteins are important because they essentially do all of the work that a cell needs to do. The directions for making proteins is held in your DNA, and then that information is sent from your DNA via mRNA to your ribosome, which then makes those proteins. And then a cell can perform all its different functions using those proteins. So maybe the protein can act as an enzyme that speeds up reactions in those cells, and then those cells working together will ultimately determine the function or behavior of a particular organism. There are several steps to build a protein. First, you need to send those DNA directions to the ribosome via mRNA. Next, the cell needs to actually build the proteins at a ribosome and then process those proteins. There are several organelles involved in processing the proteins. That will involve folding the proteins and modifying them, removing some amino acids, and then adding in some other things to those proteins. That might include adding these things called glycoprotein tags that help the cell know where to send that particular protein. Then finally, the cell will need to address and transport those proteins wherever they need to go. The organelles involved in this process are the nucleus, ribosomes, the endoplasmic reticulum, the Golgi, and vesicles. The function of the nucleus is to protect the DNA. The nucleus consists of a nuclear envelope which has little holes in it known as nuclear pores through which things can enter and leave the nucleus. The things that need to enter and leave the nucleus include nucleotides that are needed to make mRNA and rRNA, because ribosomes are actually made of ribosomal RNA. So the materials for making those rRNA, the material needed to make those need to be able to get inside of the nucleus. This is a snapshot of what happens when you unzip a DNA strand and actually copy a particular gene into mRNA. This is known as transcription. So transcription is when you produce mRNA from DNA in the nucleus. The DNA is just a template, and then it can zip back up when this process is done. The mRNA will then leave and go out and attach to a ribosome. This is this is an electron microscope image of a nuclear pore, and you can see that things can enter and leave the nucleus through that pore. Inside the nucleus, you have a nucleolus. The function of the nucleolus is to make ribosomes out of rRNA and proteins. These components will attach to one another. You'll have your ribosome, and then the ribosome will leave to go out to the cytoplasm through the nuclear pore. Your ribosomes function to produce proteins. Specifically, they are the site where you read your mRNA. It's like a recipe, and then read that recipe to make the chain of amino acids. So your ribosomes are made of rRNA and a protein, and your ribosomes consist of two subunits, a large subunit and a small subunit. It's important to note that the genes for making ribosomes, or the genes for making rRNA, are very highly conserved. They have the greatest commonality among all living things. And this is because if you were to mutate those genes, then the ribosome might not function properly, then it can't make proteins, and therefore the organism will not survive to pass that gene on to the next generation. So the genes for rRNA are extremely highly conserved. You have two types of ribosomes. You have free ribosomes, which float around in the cytoplasm, and they make proteins that are going to stay in the cytoplasm, proteins that will stay in the cell. And then you have attached or bound ribosomes, which are attached to the rough ER, or endoplasmic reticulum. And then those attached ribosomes make proteins that will eventually be sent out of the cell, or proteins that will be embedded in the plasma membrane, or attached to the plasma membrane. But the ribosomes that are free or floating around loose in the cell make proteins that stay in the cell. The function of the endoplasmic reticulum is to process the proteins that leave the cell. So we had to type it right here. So your rough ER makes proteins that eventually will leave the cell. Your endoplasmic reticulum also makes membranes and can break down some compounds. We'll learn more about that in the next couple of slides. Your endoplasmic reticulum is made of a bunch of folded membranes that are interconnected to the nuclear envelope, or nuclear membrane. You have two types of ER. You have your rough and your smooth ER. The rough ER has ribosomes embedded in its surface. Your smooth ER functions to produce lipids and then your cell's going to use these lipids to make various things, and it's going to function in metabolic processes. So there are three general functions of the smoothie R that you need to memorize. The first one is synthesizing lipids, and those lipids can consist of oils, phospholipids, steroids, and sex hormones. Phospholipids, remember, are the things that look like this. And so obviously, if your smoothie R is making these phospholipids, it can then in turn use those phospholipids to make proteins. And obviously, if it makes sex hormones, it can make things like testosterone and estrogen. Your smoothie R also functions in two types of hydrolysis. Hydrolyze just means to break apart. So hydrolysis is the process of breaking something apart. So your smoothie R hydrolyzes glycogen. It breaks it down into glucose. And then your smoothie R can also detoxify drugs and poisons. Both of these processes will occur in your liver. So certain cells are going to have lots of smoothie R. Any cell that needs to make lots of membranes, or a cell that needs to make lots of sex hormones, like male's testicles, are going to have lots of smoothie R, so they can make the lipids from which testosterone is made from. And then any kind of cell that needs to hydrolyze glycogen into glucose or detoxify drugs and poisons, such as your liver cells, will also have lots of smoothie R. Your smoothie R is essentially a membrane factory. So as you're producing more phospholipids, those phospholipids can break off almost like a little bubble and join to the plasma membrane, thus making the plasma membrane larger. Your rough ER functions to produce proteins that will be exported or sent out of the cell. So remember, you have ribosomes that are free, that are just floating around in the cytoplasm. They make proteins that stay in the cell. And then the ribosomes that are attached to the endoplasmic reticulum make proteins that will be sent out of the cell. So the rough ER helps to basically modify those proteins and then sends them to the Golgi apparatus or the Golgi body. So the question is right here, which kind of cells are going to have lots of rough ER? Well, if rough ER modifies proteins that will be sent out of the cell, cells that need to secrete or release lots of proteins are going to have lots of rough ER. For example, your cells that make insulin in your pancreas need to make lots of protein. Insulin is made of protein. And so if your cell is making lots of protein, that's going to eventually leave the pancreas cells and go out into your bloodstream to in turn regulate your blood sugar. In order to make all of that insulin, it needs lots of ribosomes and rough ER to do that. So any sort of cell that secretes lots of proteins, like your pancreas, is going to have lots of rough ER. So this is a picture of your rough ER. Basically, you have ribosomes that are attached to its surface. They read mRNA, and then you secrete the protein. So this green is supposed to represent your mRNA. The purple represents your ribosomes. The ribosomes can attach to the rough ER and then secrete those proteins. Your Golgi apparatus is like the UPS of your cell. It finishes, sorts, tags, and ships your proteins. Basically, it finishes them up and then tells them where to go by adding little chemical tags to them. Your Golgi also makes lysosomes. So cells that need to ship out lots of proteins, not only will they have lots of rough ER, they'll also have lots of Golgi. So for example, your pancreas cells, which need to secrete lots of insulin, will have lots of rough ER because that's where the insulin is made. And then they will also have lots of Golgi so that you can secrete that insulin from the cell out into the bloodstream. Also, cells that have lots of lysosomes will also need to have lots of Golgi. For example, your your white blood cells need to have lysosomes to help them break down pathogens, so they will have lots of lysosomes to do that. This is another diagram of your Golgi. Remember, your Golgi almost looks like folded membrane sacs, almost like stacks of pancakes squished together. So whenever a protein is finished, part of the membrane from the Golgi will pinch around that protein, and then it will be shipped out to the plasma membrane. This is just another image that shows you how proteins are sent or shipped around the cell. So this would be the rough ER. The proteins are being made on the rough ER by the ribosomes, and then part of the rough ER pinches around the proteins. Basically, it squishes around them like a bubble, and then this is called a vesicle. This bubble membrane is a vesicle. It carries those proteins to the Golgi. The membrane fuses to the Golgi. The Golgi can modify them, finish them up, and tag them, and then pinch off to be sent out of the cell. So this is a diagram that summarizes what we talked about or what we learned about today. The first step in making proteins is to copy that recipe from DNA into mRNA, or messenger RNA. That process is known as transcription. Then that messenger RNA will go bind to a ribosome. The ribosome can either be floating freely in the cell, or it can be attached to the endoplasmic reticulum as a rough ER. It's important to remember the difference between those two ribosomes. The free ribosomes make proteins that stay in the cell, and then the ribosomes attached to the endoplasmic reticulum make proteins that will eventually either be embedded in the plasma membrane or proteins that will be sent completely out of the cell. So once you make the proteins on a rough ER, then that protein is sent to the Golgi to be tagged via a vesicle. So vesicle is just when you pinch off part of the rough ER around the protein, and then you carry it to the Golgi. That little bubble of membrane fuses with the Golgi. The Golgi can add any tags that tell the protein where to go, and then part of that Golgi will pinch around the protein as a vesicle, and then that vesicle can fuse to the plasma membrane. Those proteins will either be embedded in the surface of the plasma membrane, like as a receptor protein or a transport protein, or they can actually be released into the bloodstream to do whatever they need to do. So this is another diagram, same idea, it's just a little bit more realistic looking. So again, you have your DNA that's in the nucleus. Okay, your cell will unzip the DNA, copy the DNA into mRNA through the process known as transcription. The mRNA will leave through a nuclear pore and attach to a ribosome. If this protein is destined to stay in the cell, the ribosome, or excuse me, a ribosome that is freely floating in the cell will synthesize the protein. If the protein is destined to be shipped out of the cell or embedded in the plasma membrane, then the ribosome that will make that protein will be embedded in the plasma membrane. Excuse me, it will be embedded in the rough endoplasmic reticulum. So after the protein is made, if it's staying in the cell, it just stays in the cell. If it is going to be shipped out of the cell, then part of the rough ER pinches around the protein as a vesicle. That vesicle carries the proteins to the Golgi. The Golgi will finish it up and tag it, add any tags that tell the protein where to go, and then the Golgi will pinch around it, carry the proteins via a vesicle, and it will fuse with the plasma membrane and release its contents out of the cell. So this is a diagram that shows you an animal cell. You should be able to trace the production of proteins from the nucleus. The DNA is going to be copied to mRNA. It'll be read by the ribosomes on the rough ER. Then the proteins will be sent to the Golgi to be modified and packaged up and tagged. They'll travel through a vesicle to fuse with the plasma membrane and leave the cell. This is a plant cell. The same thing happens. You have DNA in your nucleus. The DNA is copied into mRNA. The mRNA is read by a ribosome on the rough ER, and then that protein made by the ribosome on the rough ER is sent to the Golgi. The Golgi will modify and tag the protein so it knows where to go, and then part of the Golgi will pinch off around the protein, and then the vesicle will fuse with the plasma membrane and dump its contents out of the cell. So that's a lot. If you need help, feel free to rewind or ask questions during class.